I am very pleased to see that at least officially, no one has run screaming from the building after the first exam. So congratulations on completing that. No one dropped, so hopefully that's a good sign. I'm sorry? I There we go. I hope to have the exams graded as soon as possible. Usually it takes me about a week. During summer school, I try to get that done sooner, but especially with a Monday exam, there's not a lot of free time between now and the weekend. So I'll do the best that I can, but hopefully I'll have it back quickly. And we are on to the second part of the class, the skeletal system. As I mentioned today, we will start talking about the bone histology, bone tissue, and then we'll move on to the physiology. Up to this point in time, there hasn't been a real huge break between the lab and the lecture stuff. But now, starting tomorrow, or not tomorrow, but I guess Wednesday, we will have more of a break between the lab and the lecture part. The lecture part will be talking about all the physiology associated with bone. And then in lab, we'll be doing the anatomy, the bones and bone features. And again, rather than me just standing here going, right, this is a femur, this is the head of the femur, this is the greater trochanter of the femur, you guys are going to do that. So we'll be forming groups for the bones and bone presentations, very informal. We'll talk about that later today. And then those will start on Thursday. Notice also on Thursday, we obviously have class, but there's no assignments due. That always makes a good day for a daily quiz, so hint, hint, worry about that. But we have 4 assignments that are going to be due this section, your 3 unit reviews, 7, 8, and 9. And then there's also on the very last day of this section, a 30-point skeletal review. One of the things we're going to be doing is we're going to be grabbing the bones and becoming hyper-focused on all the little components of the bones. But it is also important to understand that these bones work together to make the skeleton. Not only do we need to understand how the bones relate to each other, but also how they form the general structures that give us our vertical axis and our structure and our form. And that is what the 30-point skeletal review is about. That's why it's not due till the end. I purposely have not made it available yet on the modules and on Canvas because I don't want you working on it yet. I know when something is worth 30 points and is graded for correctness, people want to focus on it right away. But the problem is, the point is understanding how bones and bone features go together. If you don't know bone A and you don't know bone B, and you don't know their features, then you're not going to be able to easily put them together. So it makes more sense to wait till you understand some of this material more when you're learning the bones and bone features. To be able to put it together will be much, much easier. So again, it is a short period. So I think on Thursday, I'll release that so that you have the opportunity to at least start thinking about it. But I strongly encourage you don't start working on it until you get more familiar with the bones and bone features themselves. All leading up to July 1st, which again, seems like it is rapidly approaching. It's a couple weeks away, a little over a couple weeks away. And that is going to be when you are going to have your next exam. So fun with that. All right, questions on any of that? Again, about that lab and lecture exam. Oh, I do want to talk about that. That was the other thing I wanted to talk about. Format for the lab and lecture exam will be the same for the last one. A handful of people did have some problems with the lab exam. So one of the things I had to do yesterday was convert the lab exam from having all the questions at once, which, as I talked about, is my preferred method for that, to presenting them one at a time. The handful of people who had problems, I would say from what the feedback I've gotten from Proctorio, the 2 main concerns that were consistently seen across people who had issues is either you had a poor internet connection or you did not have enough free RAM. Again, so make sure you have those things available and accessible. But because as soon as I made that change, the problems were almost completely eliminated. I think I will stick with that 1 question at a time format for the second exam. Like I said, I don't prefer giving it that way, but if it eliminates problems for the people that were having it, and there was a fair amount of that had it, then I guess it's worthwhile to do it that way. But other than that, you've now had a lab and lecture exam, and the exact same way I wrote that exam is the exact same way I'm going to write the next one, so you know what to expect. So there are no more surprises. So that is the excellent thing about that. All right, questions on any of that? All right, silence. Excellent. Love my silence first thing in the morning. All right, let's go ahead and get started then. Like I said, we are on to our second organ system now, and the second organ system, oh, I don't have my chat window up, is the skeletal system. Again, skeletal system comes from the word skeletos, which means dried up, because usually when we see a skeleton, that's how we see it, right? You know, good old Yorick holding his head there as we're doing that. However, we think of bone as this very dried up, you know, inert material. But as we've already kind of hinted at, bone is an incredibly dynamic tissue. As we talked about, you break a bone and it heals, right, in 4 to 8 weeks usually, or something like that. So it is an incredibly dynamic tissue, changes constantly, and there's a whole lot of physiology going on in it. Now, the skeletal system is an organ system, so it must involve organs. And what are the organs of the skeletal system?
Bones.
The bones themselves.
Are bones, though, really organs?
Yes, because they're made of multiple types of tissues.
Absolutely, right? Again, as we talked about, a bone absolutely is an organ. It is made up, yes, primarily of bone connective tissue, but there are also blood vessels in it, so all the tissues associated with that. There's other connective tissues associated with it, nerves in it, all sorts of other structures in there as well. So absolutely, it is an organ. And how many do you have of these organs?
106.
206, that is the magic number that everybody always gives us. But really, there's a huge caveat with that. There are 206 named bones, right? That means that there are 206 bones that pretty much everybody has inside of their body in the exact same location. And so for consistency's sake, we give them all the same names, like the humerus or like the second rib or something along those lines. However, every single person in this class has dozens of additional bones. These are small irregular bones that have fancy names to them, but they differ from person to person. Some of them are located in your skull. Some of them can be located within your joints, for instance. I may have 3 in my wrist. You may have 5. Somebody else may have none in their right wrist, but may have 2 in their left wrist. And because of this, these dozens of additional bones don't have specific names because they're not consistent between person to person. However, what is consistent is that everybody has them. So the actual number is far, far above 206. However, we focus on the 206 name bones because those are the ones that are consistent. And in fact, you will be responsible for identifying all 206 name bones in the human body. So absolutely, bones are organs. We've now talked about the numbers. And of course, the study of bones is the field of osteology. All right. Oops. We've talked about skeletal system before, and when we have, we've primarily focused on these 3 main functions, right? Bones provide support, they provide our structure, they give us our vertical axis, right? And that's a huge part of it, right? Again, yes, they provide protection. Some bones provide protection, like our skull provides protection for our brain, but how many bones do I actually have in my hand? Anybody know?
36.
36 is not a bad guess. Not quite correct, but it's a lot. Absolutely, you are correct in that it is a lot. And notice none of them are providing any protection, right? They're providing support and they're providing movement, right? That leverage, those levers for our muscles to pull on. But in fact, there are actually 6 functions to our skeletal system. There are these 3 that are the common ones we think of, but there are 3 other very important functions of the skeletal system. The first is hematopoiesis. Hematopoiesis is always a fun word to say and a fun word to have to spell. Anyone know what that actually means? Anyone want to hazard a guess what hematopoiesis may involve? You know this even if you don't think you know this.
Is it the production of blood or red blood cells?
Absolutely, and not just red blood cells, but the production of all blood cells. Red blood cells, the white blood cells, and the platelets. Or, as we learned for the last exam, our erythrocytes, our leukocytes, and our thrombocytes. Now, this occurs because bones have, or contain, I should say, a pluripotent, right? Remember, pluripotent means that it can produce multiple things. A pluripotent stem cell that is known as a hemocytoblast. And that hemocytoblast is able to produce any of the cells associated with our blood. Red blood cells, there are actually 5 main types of white blood cells. And if you remember, as we talked about, platelets aren't actually cells, they're cell pieces. So they're actually able, this hemocytoblasts can produce a cell called a megakaryocyte, which is not one you need to know, but that megakaryocyte is how we make our platelets. Now, the question becomes, where does this hematopoiesis take place? Where in our
central canal.
Okay, that is a good guess. It definitely is within a space, but we need a larger space than the central canal. Remember, the central canal is going to be small enough where pretty much just the blood vessels and the nerves and maybe some lymphatic vessels are going to be located. We need a bigger space. Is there a bigger space than central canals in a bone?
The marrow?
Yeah, absolutely. Many bones have some hollow spaces in the middle that contain marrow. However, as it turns out, bone marrow comes in different flavors. So which of our bone marrow, if I actually know what I'm spelling, is actually responsible for making our formed elements?
Red bone marrow?
Yeah, absolutely. Red bone marrow. Absolutely. Red bone marrow, located typically in 4 of our bones, is where we find these hemocytoblasts that allow us to make more blood cells, all of our blood cells. Bone is also vitally important for the storage of minerals. One of the primary minerals that is stored in our bone is calcium. In fact, something like 99% of all of our calcium is stored in our bones. But it isn't just calcium. There's things like phosphorus and magnesium and manganese and all sorts of other minerals that are stored there as well. And notice I didn't just say mineral storage here. We said mineral homeostasis. Because again, I know every single one of you before class today had a nice, well-balanced breakfast that involved a large glass of milk or a big slice of cheese or something like that, so that you were ingesting lots of calcium into your body to help keep your body functioning properly, because as we know, calcium makes cells do wonky things, so we have to have the right amount of it in our blood. But there are actually people out there who will have a donut and a Diet Coke for breakfast. And if that is the case, are they necessarily getting the calcium they need from outside of their body? No. So what will their body do then in those instances?
Release some of the stored calcium?
Yeah. If it comes to a choice between having big, strong, and healthy bones and having enough calcium in your blood, which one's actually going to win?
Strong bones.
Actually, the calcium in the blood. You were right the first time. The calcium in your blood, absolutely. Having that calcium in your blood is so vitally important for cells to function properly, that if we have to hurt the bones to make sure that we have enough calcium in the blood, our body will actually do that. So really important storage area for these minerals so that they can do that. And again, key question, where are all of these minerals stored? Where's the calcium?
Is it just inside the matrix?
Yeah, there you go. Absolutely. Perfect. Right. Remember, as we talked about, the matrix of our bone connective tissue is primarily those calcium crystals. Absolutely. Perfect. Excellent. So stored in the matrix of our bones. But it turns out minerals aren't the only things that are stored within our bones. We also store triglycerides. Now, the first and most obvious question then is, of course, why? Why do we store triglycerides in our bones? Why do we store triglycerides in general?
Energy.
Energy, absolutely. All right, as we talked about, bone is very dynamic, so having those triglycerides nearby can help with some of the dynamic processes. But there's another important reason as well. We're almost at summer. Well, actually, I guess we are at summer. We've already had the summer solstice. But I always think of this more occurring around the first of the year, right? When everybody's making their New Year's resolutions. Everybody has the commercials on TV to try to get you into your bikini body shape. And one of the things they like to do is they like to show you a pound of muscle and a pound of fat. And what's the difference between a pound of muscle and a pound of fat? Both weigh the same, but what's the difference when they show those to you on those infomercials and stuff like that?
The fat is way bigger than the muscle.
That's way, way bigger, exactly, because it is a very light material. So the other advantage of putting these triglycerides inside of our bones is to help with weight and balance, to help to lighten the weight of the bone, right? If it was solid bone, these things would be super heavy and hard to move around. So it helps with our weight, it helps with our balance by having those triglycerides inside of our bones. And of course, where are those triglycerides located?
In the matrix as well.
Matrix is a good guess, but I'll give you another hint. Notice up here, we didn't just say bone marrow. We actually talked about a specific type of bone marrow. Implication, if there's a red bone marrow, there must be some other type of bone marrow. And what would that other type of bone marrow be?
Yellow bone marrow.
Exactly. Yellow bone marrow basically is red bone marrow that has filled up with adipose. So putting all that adipose in there makes that bone marrow less functional for making red blood cells and white blood cells and platelets, but it does help to lighten and maintain the balance of our skeletal system. Excellent. So there you go. With that all together, we now have our 6 main functions of our skeletal system. Questions on that? All right, excellent. So let's talk about bone. As we know, bone organ is primarily made up of bone connective tissue. And again, if that starts to get too confusing, then remember we also have the option, instead of bone connective tissue, of using the term osseous tissue. And again, osseous tissue is a connective tissue, so we know it is made of cells and matrix. So let's talk about the cells. There are 4 primary types of cells that are found in bone connective tissue. And for this, let's switch to our whiteboard. Do some quick drawing first. I'll start easy with just some big generic dino bone. There we go. We have our dino bone. And again, there's a couple things we know about it. As we were mentioning, there's going to be a chewy nougat center, or at least a hollow center in the middle. And that hollow center in the middle, as we talked about, is where we're going to find either the red or the yellow bone marrow. Then we have our big white uniform looking, at least to the naked eye, bone connective tissue. But there's some other anatomy we want to talk about as well. For starters, as it turns out, there is a dense, irregular connective tissue that wraps around the outside of the bone. In fact, this is very, very similar to what we saw in cartilage. Cartilage also has a dense, irregular connective tissue that wraps around the outside of it. Clearly, this isn't the only place we have a dense, irregular connective tissue. So that dense, irregular connective tissue that wraps around cartilage, what did we call it based on its location? Anyone remember from the last exam?
Was that lamina propria?
No, not a good guess. That is a good guess. That is definitely a based on location term.
Mesothelium?
Closer, that is the simple squamous epithelial tissue, also a based on location name. But no, this one went around the perimeter of the cartilage. What did we call that?
The perichondrium.
Perichondrium, excellent. Well, this one happens to do the exact same thing, only around bone. So not surprisingly, based on its location, we call it a periosteum. And it turns out there is also a dense, irregular connective tissue that lines that cavity on the inside of the bone as well. This also is a dense, irregular connective tissue, but this one is on the inside of the bone. And because it's on the inside of the bone, we can't call it a periosteum. Have any guess what we might call it when it lines the inside of the bone?
Endosteum.
Yeah, exactly. Endosteum, right? Endo means within, so endosteum. Excellent, 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 excellent. I make a point of mentioning this because here's what I want to do. So let's first cheat and move this over to this side instead. What I want to do is take a cross section through a little piece of this bone. And we'll put it over here to the side so it's not too much in the way. But if we were able to do that, what we would see is here. So let's go back to black. Here is the corner, so the outer surface. Here's where that cavity is located, and I'll draw a little bit of them on the outside. We know the periosteum is located here, and the endosteum is located here. So does that kind of make sense how I've taken this image and taken a little bit of a cross section here and put it over here to make some sense of it?
Yeah.
Excellent. All right, so now I can erase all of this. And instead, what we can do now is we can focus on the 4 main types of cells that are found in our bone connective tissue. The first of these are what are called an osteogenic cell. These osteogenic cells are unipotent stem cells. Remind me again what that means?
They can only make one specific cell.
Exactly. They're able to, their stem cells divide, make new cells, but when they make new cells, they basically form only one thing. And what's special about these osteogenic cells are they're the only bone cells that are capable of dividing. Oops. These osteogenic cells are mostly found under the periosteum, but also can be found under the endosteum. So again, some of these, and we'll just make them blue dots for right now, are going to be found here. And I'm going to have to cheat for this a little bit and extend out. We know a periosteum is going to be in direct contact with the bone, but I'm going to cheat and stick it out a little bit more so that we have a little bit of room to play. And then same thing here. Some of these osteogenic cells will be located here under the endosteum as well. Now, these are unipotent stem cells. So when a unipotent stem cell divides, it produces one and one thing only. Oops. And the one thing that it produces is the immature bone cell. And of course, as we know, the immature bone cell is an osteoblast. And that is our second bone of the connective tissue. Osteoblasts, of course, are immature cells. And as we know, what do immature cells do?
Build the matrix.
Make the matrix. Excellent. Now, in the case of osteoblasts, the way they do this is they take the calcium, they take the phosphate, they take the manganese, and they basically deposit it onto collagen fibers. So basically, they take the minerals and they deposit it on collagen fibers. This is a process called mineralization or ossification, where they're making the bone. And they make the matrix. And so they form this matrix around themselves. So this one here is going to make a little bit of matrix, and then a little more, and a little more, and a little more, and a little more, and a little more, until suddenly it is completely surrounded by matrix. Now, of course, when it surrounds itself with matrix, is there any place else to put matrix now? If you're building a sand wall and you've completely surrounded yourself with a sand wall, can you make any more sand wall? No.
Yeah.
So at this point that it's surrounded itself with matrix, it can't make new matrix anymore. all it can do is maintain the matrix around it. And what kind of cells just maintain matrix?
Osteocytes.
There you go. So notice these cells then mature into osteocytes, which happens to be our third type of cell. Notice I said that there are four main types of cells in bone connective tissue. But really, if you think about it, this is just the exact same cell at different stages of its life. It starts as a unipotent stem cell, divides to produce a new cell. It is an immature cell. That cell makes the matrix, surrounds itself with matrix, until it is a mature osteocyte. Again, these osteocytes are the mature cells. Their job is to maintain the matrix. And as you notice, like I said, in many ways, it's about the location of these. In fact, if this bone breaks and an osteocyte is released from its space, and what would this space be called again?
Lacuna.
There you go. Perfect. Excellent. This space right here would indeed be called a lacuna. If that osteocyte is released from the lacuna, it will actually regress back into an osteoblast to make more matrix again. So it really is kind of about just the location of them. Now, there is some interesting characteristics about this osteocyte. Now, we kind of remember, hopefully, where these osteocytes are located. So let's do a little bit more vocabulary here. We know within this matrix of our compact bone, what was that organizing structure of the compact bone called again?
Central canal.
Well, close. The central canal is the space at the center of that organizing structure. But what's the organizing structure itself called?
Osteons.
Yeah, an osteon, absolutely. So we have a central canal at the center of our osteon. So let's write this. And as we know, that central canal is going to be where the blood vessels are going to be located, and the nerves and maybe some lymphatic vessels. And then, as we also just mentioned, let's go ahead and change the color of this so it's easier to know what we're talking about. There's a lacuna. Obviously, those are not drawn to scale, but you get the idea. We have the lacuna there that we were talking about. And that lacuna contains the osteocyte. Now, one of the interesting things about these osteocytes, and I'll just draw an osteocyte here to the side. Oh, no, hold on. I want that to be purple. So that over here, I can draw an osteocyte. When the cells mature, no, not enough. Not only do these big blobby osteoblasts elongate so that they can fit in that lacuna, but they get these big, long processes that stick out of them. The reason for this is some osteocytes are going to be closer to the central canal and be able to easily get oxygen and nutrients. And so what'll happen is these cells will actually put out these processes where they can form gap junctions with the nearby cells. And in that way, they can pass nutrients and oxygen and other materials back and forth. Right? Because again, the matrix is a hard crystalline matrix. Yes, there is water in it, but it can be hard to move the substances we need through it. So as these cells mature, they narrow and elongate. They narrow and elongate. And as they do that, they form long processes to form gap junctions with other osteocytes. So they can be constantly maintaining the matrix. All right. So those are our first 3 cells, but we have one more we need to do. This 4th cell is what we call an osteoclast. Notice osteoblast, and one of the ways I always remember this is the osteoblast has the B for build, whereas our osteoclast has a C for chew, because that's what osteoclasts do. Osteoclasts are the cells that are responsible for breaking down bone matrix. These osteoclasts have some very interesting characteristics to them. They are actually more similar to white blood cells. Than they are to the osteogenic, osteoblast, osteocytes that we were talking about. So this isn't some 4th stage of the cell. This is a different type of cell with a different anatomy. This cell has multiple nuclei, and it has a big, huge, what they call ruffled border. I'll draw it in a second so you can see what it looks like, but we need to know where to draw them, and that is because most are found under the endosteum, where some can be on the periosteum as well. Most of them are going to be located here under the endosteum. So again, it's a large cell with a big, huge ruffled border. This big, huge ruffled border gives it a large surface area. This cell has multiple nuclei in it. And of course, as you know, nuclei are where we keep the instructions for making proteins. So not surprisingly, this cell makes a large number of hydrolytic proteins. And of course, those hydrolytic proteins break bonds. And so it is their responsibility to break down the bone matrix. And of course, when you break down bone matrix, what happens to all the calcium and the other minerals that are being released?
Gets put back in the blood.
There you go. So it releases calcium and other minerals into the blood. Of course, conversely, these osteoblasts that are building are, of course, taking calcium and minerals from the blood, really from the interstitial fluid, but of course the interstitial fluid gets it from the blood, the same way that the osteoclasts release it into the interstitial fluid, but it essentially then immediately goes into the blood. So we pull it out of the blood to deposit it into the bone, or we break it down from the bone to release it back into the blood. Now, of course, as you can see here, I've done a truly amazing job of drawing and identifying these things, but let's look at the pretty pictures from your textbook that do the same thing. Here is that unipotent stem cell. Again, remember, mesenchymal stem cells are the pluripotent stem cells that form all connective tissue cells. So that mesenchymal cell divides, and when it divides, it forms an osteogenic cell. Let's cheat and move this up here. And that osteogenic cell is a unipotent stem cell that can divide to form one thing and one thing only. These are found both in the periosteum and the endosteum. And again, being unipotent, when they divide, they form one thing and one thing only, osteoblasts.
Professor, quick question.
Yes.
So when it divides, it makes two osteoblasts, or does it one stay as the stem cell and then one osteoblast?
Depends on the type of growth that's going on. If it's healing or something like that, both cells could potentially become osteoblasts to then help to repair the damage. But usually you've absolutely got the right idea. Typically when these stem cells divide to produce two cells, one stays a stem cell so that we can continue the repair process later on, whereas the second one then becomes whatever the new cell is that we need. All right, excellent, great question. All right, osteoblast, as we talked about, B for build. These are the ones that make the bone matrix, take the calcium, take the manganese, take the phosphates, and deposit it on those collagen fibers. Like we said, it's the process we call ossification. It's the process we call calcification, right? It is what we call osteogenesis, the making of bone. Once they surround themselves with bone, they become osteocytes. And again, this is the picture from your textbook. It's not the best picture. You can see the cell is narrowed a little bit, but it has those processes that come off. Here's a picture from a different textbook that I think it does a little bit of a nicer job of showing this nice elongation of the cell, narrowing of the cell, and those big, huge processes that stick out where it's going to be able to form gap junctions with some processes of another cell so that they can pass materials back and forth from each other. And again, osteocytes, being the mature cells, are responsible for maintaining the tissue. Here, we see that fourth cell, that funny looking osteoclast, C for chew, mostly found on the inner lining of the endosteum. Multiple nuclei to make lots and lots of proteins, big ruffled border so that it has a huge surface area, so that it is able to break down that bone matrix and release it into the blood. And that process of breaking down bone matrix and releasing it into the blood is a process we call resorption, or basically reabsorbing those minerals back into our blood. Very important for helping us to regulate calcium levels, because like I said, if it comes to a choice between, right, if we're not getting the calcium we need from our diet, we have to have and maintain important levels of calcium, and it's either let calcium levels drop or break down our bone matrix. And given a choice between those two, your body will pick breaking down your bone matrix every time. All righty, questions on that? Those are the cells of our connective tissue bone. Any questions on that? All right, let's talk about the matrix. Of course, as we know, matrix is made up of two components. Let's actually do that here. Our matrix is made up of ground substance and fibers. That is true for all matrices and definitely true for our bone connective tissue. As we also know, ground substance is mostly water. And sure enough, again, we think of bones as these dried up, shriveled up things, but there is a large amount of water. In fact, the matrix is about 25% water. And as we talked a little bit about in a previous class, I'm not sure exactly which class it was we were talking about it in, but remember, bone is also about 25% collagen fibers. Remember, we made the joke about dropping babies and having them bounce and grandma's rolling off of changing tables and all of that. Our bone has collagen fibers in it, a lot of collagen fibers, giving it its tensile strength. Usually at this point when we're in the classroom, I actually have some bones that I get to hand out to you. Some of those bones have been baked in the oven. And when they've been baked in the oven, basically the water has been evaporated away from them. The collagen fibers have been broken down. And all it is the hard crystalline matrix, right? That hard matrix that we have of the bone, and it's rigid and it's solid. Whereas we have some other bones, and again, this is something you could do for yourself. When next time you eat a chicken, take the two leg bones, one of them, strip, put in the oven, bake at about 250 for about an hour, and you will destroy all the collagen, remove all the water, and it'll be hard as a rock that you can pound on the table. The other one, take and put in a jar of vinegar and leave it in there for a couple weeks. And when you leave it in that jar of vinegar for a couple weeks, what happens is that the minerals are broken down by the vinegar. And all you're left with is the bendy collagen fibers. And so this is when I pull out the chicken bone that actually bends like it came from rubber chicken, and everybody oohs and ahs as a result of that. And I pass around and you get to play with it as well. But again, we're not in the classroom, so we don't get to do that. It's what gives the bones the flexibility and the give. We talked about bouncing babies, but also the fact that you can jump, that you can twist and you can turn. It has that resistance to bending and flexibility. We also talked about the third component, this organic calcium minerals, primarily made up of calcium phosphate, but also calcium carbonate and manganese and some things along those lines, is a crystal called a hydroxyapatite crystal. This hydroxyapatite crystal is the hardest substance produced by the body. And not only do you find it in your bones, but it also forms the enamel on your teeth. However, your teeth are not bones. And the enamel there is similar because it has the hydroxyapatite crystals, but what it doesn't have are collagen fibers. So as we talked about, when you're eating that bowl of clam chowder and you bite down onto that clam shell, with no collagen fibers, that enamel of your tooth doesn't have any flexibility, doesn't have any give. So while it's very, very strong, like china, it can also be brittle. It has no give, and so that's how you chip that tooth on that clamshell. Bones, you can jump up and down, right? You can twist and you can run. You can put stress on them, and they have some flexibility. They have some give because of those collagen fibers. All right. Questions on that? All right, excellent. So obviously we have to go from the rubber bouncy ball collagen fiber bones of a baby to the mature hard crystalline bones of you as an adult. That process is called calcification or mineralization. And like I said, it occurs by our osteoblasts taking those minerals and depositing them onto the collagen fibers. Without those collagen fibers, you don't get crystalline bones. Now, in this process of calcification, and I mentioned this briefly when we were going over the connective tissues, is there's not one but two specific types of bone connective tissue. Those two specific types, anybody remember what we called them? There you go. Samuel jumped ahead, knew where I was going with it. Excellent. Spongy and compact bone. Excellent. Compact bone is the majority of our bone. It is about 80% of our skeleton. And compact bone is also the superficial bone. So again, I have to work on my examples for this because normally this is when I grab Bob and roll him out into the middle of the room. We've got all these skeletons hanging around inside the room. And when you look at him, you look at a skeleton, you look at a bone, typically all you see on the outer surface is that smooth, uniform looking compact bone. To the naked eye, it looks white and smooth. When we look at it under the microscope, we see it's made up of those osteons that we've talked about. And that is indeed how we define compact bone. It is made up of the organizing structures known as osteons. And the second type is what is known as cancellous or spongy bone. And for me, spongy is much easier to spell and easier to remember, so that's the one that I always use, but both are appropriate anatomical terms. It gets the name spongy because much like if you grabbed your kitchen sponge and looked at your kitchen sponge with the naked eye, right? It's made up of all of these irregular points and processes, right? There's all these irregular points and processes that don't have one just uniform shape. Remember, as we saw, that osteon is a bunch of, right, concentric circles around the midpoint. It had a very distinct structure to it. These irregular points and processes don't have a single uniform shape to them. They look like and appear like a kitchen sponge, and that's where it gets its name. Right, spongy bone has these irregular points and processes that we have to give some name to because anatomists must name everything. So they refer to them as trabeculae. Of course, trabeculae is the singular, or trabecula, I should say, is the singular. Trabeculae is the plural. And like I said, spongy bone is deep and also only makes up about 20% of the bone. Here, just like I drew on the board before, we have a partial cross section through a bone. Notice, just like we did before, we have a periosteum on the outer surface. There is an endosteum that would line the inner surface as well. But notice also, lining the inside of our bone are the irregular points and processes of our spongy bone. One of the places we find spongy bone is lining these cavities inside of our long bones, like this one here. And then here, change colors, we have our compact bone, and they've even pulled out one of our osteons for them so that we can, so that we can look at and identify and see the anatomy of that. All right, let's leave here. Questions on that? Now, as you've seen from your handout, hopefully you've looked at your histology handout, there is a lot we need to take in on this. This is actually probably a good time to point this out as well. Obviously, I'm certain that you took a good 15-20-minute break after the exam, and then of course, dove right into the new material for this section. But on the off chance that you didn't, when you go to the section for the skeletal system, we have the places for the assignments, we have our lecture slides, we have our handouts, 30-point review, which again is not ready yet, but there are a couple other really important things here. And of course, the first one is your skeletal system histology handout. Notice the histology is not anywhere near as elaborate as it was for the connective tissues and epithelial tissues, but there is still a fair amount of histology. Well, not a fair amount, but there is still some. You absolutely positively are going to be responsible for this under the microscope, and we will look at this under the microscope, but you will also notice that under the modules, there are some pictures of some, under the lab handouts are some pictures of some of the models from the classroom. This is a model just like the drawing we were looking at, just like the drawing I was showing you before, where you have your periosteum on the outer surface, endosteum on the inner surface, compact bone and spongy bone. And you will notice that this illustration shows them all from all points of view. We're in the inside of the bone here. Here's the outside of the bone from the top and everything like that. I guarantee you this model is going to be on your lab exam. So when we talk about the histology, you'll be able to identify all those things on the list on here as well as on the microscope. And in fact, some of the things here are easier to see on this bone model than they are on the histology slide. So make sure you're familiar with both of those. All right, now there is a fair amount of histology on that, and I do want to briefly go through it together, but it is a lot of vocabulary, a lot of information, and so it might be worthwhile for us to, even though it's a little bit early, go ahead and take our first break now so that we can caffeinate and we can prepare and we can get ready to go over all this anatomical terminology or histologically terminology. So that's what we're going to do. We'll go ahead and take our first break now, come back and dive into all of this vocabulary. All right, any questions on that before we take our first break? All right, we will restart in 15 minutes. So it looks like that is going to be, what is that? 9:12. And I will start the recording at that point. All right, I will see you guys in 15 minutes. Let's talk vocabulary. Any questions before we get started? So, like I said, bone tissue comes in two specific types, compact bone and spongy bone. And again, there are a couple big key distinctions between them. The first is the organizing structure. And the organizing structure of compact bone is this thing we call an osteon. Now notice, conveniently for us, they have pulled one of the osteons out here and pulled one out entirely when we look at it this way. We can see a couple of characteristics about it. First, as we've talked about already, we know at the center is our central canal, containing things like nerves and blood vessels, and even some lymphatic vessels can be found in there. It is also made up of layers of matrix. And again, we couldn't get away with something as easy as just a term like layer of matrix, so we have to have a specific term for them. And the specific term for a layer of matrix is a lamella, lamella being the singular, and of course, lamellae being the plural. Notice one of the things about the lamellae is the orientation of the collagen fibers. Notice each layer of the matrix has, each lamellae, layer of matrix has collagen fibers at opposite orientations. It's what gives the bone the ability to be able to flex, the ability to be able to give. All right. So here we see what it would look like in a cross section. Here we have the central canal. Here we see the layers of matrix. And notice when we look closely, we can see one other thing as well on this. Remember, we've talked about before how this space here is the lacuna, the caves where our osteocytes, the mature cells, live. But if you notice closely, I'll use the light blue. We can also see all these dark lines that are radiating out of the lacunas. Remember, one of the things that we talked about with the osteocytes is that they have long processes that allow them to reach out to the cells next to them so they can form those gap junctions so that they can get oxygen and nutrients and pass them back and forth. Here we see another great example of these interconnected tiny little canals where the processes are located. And in fact, that's what they're called. They're called tiny little canals. And the term for a tiny little canal is a canaliculi. So these little small spaces radiating off of the lacuna are the canaliculi, where those osteocytes contain those long processes. Now, as I mentioned, a layer of matrix is known as a lamella. But as we look at our compact bone, we see that it is comprised of different types of lamellae. For starters, as we've mentioned, we have that organizing structure, the OS, that is the osteon, made up of layers of matrix that wrap around the central canal, forming these concentric circles around the central canal. And what might be a good name for a layer of matrix that wraps around, forming concentric circles, forming our osteon? I'll give you a hint. Seriously?
Concentric lamellae.
There you go, concentric lamellae. Excellent. So these concentric lamellae form the osteons. However, notice that all layers of the matrix are not forming osteons. Notice on the outer surface of the bone are layers of matrix that go, dare I say, around the entire circumference of the bone. Obviously, we just have a single wedge of the bone here, but if we had the entire bone, you would see that it would go around the entire circumference of the bone. And guess what we would call a layer of matrix that goes around the entire circumference of the bone?
Circumferential lamellae.
Excellent. And finally, and I'll change colors to make sure to emphasize this, notice that there are some layers of matrix that don't meet either criteria. They're not going around the entire circumference of the bone, but they're also not forming the osteons. After all, osteons are circular structures like straws. If I were to make a tissue by holding a bunch of straws together, there'd be a lot of little spaces in between the straws. And we would need to fill these spaces in between the straws with little irregular layers of matrix in between the osteons to kind of fill out the tissue. And what do you think we call these irregular layers of matrix that are found inside of the connective tissue?
Interstitial lamellae.
Interstitial lamellae. So notice as we said, there are two key characteristics to compact bone. The first is that its organizing structure is the osteon. And the second characteristic of compact bone is it is comprised of all three types of lamellae. We have the concentric that form the osteons. We have the circumferential that wrap around the entire circumference, so let's not use the same word, are long straight layers around the outer surface of the bone and our interstitial lamellae, which are irregular in shape and fill in the spaces. So all three types of lamellae make up compact bone.
Professor, the white material that's in between the osteons and the interstitial, is that
Just artistic.
sort of?
It's just an artistic representation to help to differentiate and show us. When we look at this under the microscope, you're not going to see that type of divisions in between the two. It's just our artists trying to really help to emphasize the different types of lamellae there. So great
Okay.
question. I appreciate you asking that. But yeah, no, when we look at this histologically, it doesn't look like that at all. All righty. So there you go. We have, again, the circumferential, concentric, and interstitial lamellae, all forming our compact bone. All right. Now notice one other thing as well. As we know, compact bone is made of osteons. And also notice the osteons all pretty much run parallel to each other. Again, there's some slight variation, but mostly run parallel to each other. And that means their central canals are also parallel, which is absolutely fine and acceptable, except when we remember that there are blood vessels in this. And obviously blood needs to be able to get both into and out of these central canals. So not surprisingly, not only do we have central canals that are at the center of our osteons, but connecting the central canals are some spaces that are known as the perforating canals. Our book does a great job of showing this as perfect right angles. Of course, are they going to be perfect right angles to each other? No, but if we go to that picture from the model in the class, notice here we see the same things. Notice here our osteons are running up and down. And so when we look, oops, there we go, at our cross section here, we can see some of the central canals that have been cut through when we cut our bone. But notice also, we can see that there are perforating canals that go from the outside of the bone towards the inside of the bone and interconnect these as well. What's nice about the model on this side is that it just shows the spaces, because the central canals and the perforating canals are spaces. But notice when we look at the other side, you can see where the blood vessels would actually be inside these spaces. And notice also when you look closely, you can see that our osteocytes here have the little processes sticking out of them. So that if you look really carefully over here, for instance, you can see the lacuna, and you can see how they've given us some of those canaliculi coming off of that as well, those spaces where those processes would be. Excellent. Questions on that?
When your bone breaks, does it rupture the veins and the arteries, and when they put it back together, do those fuse back together, or are they just kind of lost to the streets?
New ones grow into the area. So, and in fact, after what, like we did with skin, once we learn about bone and how it functions, we will learn about actually how it repairs. So we will actually talk about the repair process. But the short answer to your question is yes, these blood vessels are damaged, but they will be repaired or replaced. Yep. All right. Now, one last thing about the central canals and perforating canals. If you look in some textbooks, they still refer to them as Haversian canals and Volkmann canals. because good old Bob Haversian and Bob Volkman were the first ones who described these and identified these. And so you may still see them. Haversian is the term for central canal. A Volkman is the term for perforating canal. You are welcome to use them if you want. It is completely unnecessary, but if that's the way you learned it back in high school or something like that, and those are the terms that are stuck in your head, you're welcome to do that, but it isn't going to be necessary. Central canal and perforating canal, I think, does a better job of explaining it. But as always, you're always welcome to use any appropriate anatomical term.
Will you use those on an exam?
No, I will not use those on the exam because they're so antiquated. If there weren't still textbooks that use them, I wouldn't even be mentioning them. But unfortunately, there are, so I have to briefly mention it. But no, I will not use those terms. All right. So that is our compact bone. Now we get to talk about our spongy bone. And I guess actually, if we think about it, there's really three characteristics to compact bone. It's the outer layer, right? It's found on the outer surface. Again, the organizing structure is our osteon. And as we talked about, it uses all three types of lamellae. So those were characteristics for our compact bone. So not surprisingly, those are ways we can compare spongy bone. Spongy bone is going to always be deep. That's not how you spell deep. There we go. Away from the surface, like the inside of this long bone that we see here. Its organizing structure is just this vague thing known as a trabeculi, which, as we talked about, are just irregular points and processes. So as we look at this, it's not like there's one pattern that repeats over and over again. There's all these short, branching, irregular shapes. Some are curved, some are pointy, some are thick, some are thin, right? And they're just at all these different orientations, giving it that spongy appearance. Now, let's think about this then. If we were to form these types of irregular structures, when we are going to use, because it's still bone, so we still use layers of matrix. Because it's on the inside of the bone, is it going to have a straight layer that goes around the entire circumference of the bone?
No.
No, so we're not going to use circumferential lamellae. Also, there are no osteons. There are no central canals. So are we going to have perfect circles inset on other circles, making these trabeculae?
No.
No, so there's not going to be any concentric lamellae. So what that means, when we make these trabeculae, they are only formed by our interstitial lamellae. So unlike compact bone, which will have all three types, spongy bone only has one. And I think I have a nice picture that shows this. They kind of show us a little bit of this, and I'll use the highlighter here, down here where we see some layers of matrix, but let's take a little bit of a closer look. Notice here with our cross section, it is still a layer of matrix. Notice we still have lacunas. Those lacunas still have canaliculi coming out of them. So it is still a layer of matrix with a lacuna with canaliculi, but they're not a circle around a central canal. They're not going around the entire circumference of the bone. So they're made of interstitial lamellae only. And notice one more thing. This picture has peeled it off so that we could see it a little bit easier. But notice there would be an endosteum around this. And this endosteum, notice, is housing the immature cells, housing the osteoclast on the surface of this. And this we see nicely when we go back to our model. Notice in our model, here from this view, we can clearly see our periosteum on the outer surface. And since we're here, you know what? We'll go ahead and talk about this. Notice there are all these collagen fibers that are penetrating deep into the surface of the bone, helping to hold this periosteum on its outer surface. As we've talked about, we know the periosteum houses the immature cells, but it also stabilizes the blood vessels. It also protects the bone, and it also acts as an attachment site. This is where the tendons and the ligaments will connect. And remember, as we talked about, when you pull on that tendon and ligament, you're actually more likely to break the bone than you are to tear the tendon. And one of the reasons for this is these collagen fibers from the ligament, from the tendon, connect to the periosteum, and some of those collagen fibers penetrate deep into the bone, holding them in place. What do you think these long penetrating collagen fibers are known as? How would you identify them if I pointed an arrow at them on the exam? If only I had a list of the histology I was responsible for on this exam, I might be able to figure it out. No, we'll get to the inner layer in just a minute. We're still on the outer layer here. Do we have a list of the histology we're responsible for? A study guide, dare I say, for the histology on this exam?
Yeah, trying to get it. Sharpie's fibers?
There you go. Exactly. Because good old Bob Sharpie was the first one who identified them. They are also known as perforating fibers. So those fibers from the periosteum that hold it and anchor it in place are known as the perforating fibers or Sharpie fibers. Both of those are acceptable terms. Now, notice when we go back to our picture, so again, here is that dense irregular connective tissue. Here is that periosteum on the outside, and we can see a little bit of the perforating fibers from the top. But notice when we look at the inside, here we see the trabeculae. Remember, spongy bone lines the inside of these bones. So here we're seeing the trabeculae, the irregular points and processes. And here, the pink stuff, the purple stuff on the outer surface is the endosteum. Notice the endosteum being on the inside isn't going to be protecting the bone. It isn't going to be an attachment points for tendons and ligaments, but it's still going to house immature cells. It's still going to house the osteoclasts, and it's still going to help to stabilize the blood vessels in place. So when we look at it from the top, again, here is the periosteum on the outer surface, and here is that endosteum on the inner surface. So that picture showed us the immature cells, showed us the osteoclast, showed us a trabecula, but it didn't show us the endosteum. So like I said, the only thing that was missing from this picture is they didn't give us the endosteum around it. However, if we cheat, and it's not really cheating, but if we go back a slide, oops, hold on, here we go. Notice here, they do show us the endosteum wrapping around it. So this view, the lower magnification view illustration, they show us the endosteum. But then when they give us the close-up view of that cut section, they just show us the cells and the layers and those. All right.
So will there be the osteoblasts and on the periosteum and the endosteum, it's not going to be in the middle of the actual bone?
Correct. Yes. So the immature cells, the osteogenic and the osteoblasts, are only on the outer surface. So let's actually go back here. Right. So immature cells are only going to be found here under the periosteum and here under the endosteum, because notice all the cells in the middle are all completely surrounded by matrix. So these are just going to be the osteocytes in all of these lacunas. So these lacunas contain all the mature osteocytes in the center of the bone, and then all the immature bones in the immature cells and the osteoclasts are just found in the periosteum and in the endosteum. Yep. Which, again, if you think about it, makes sense. It's why we said that those osteogenic cells are the only ones that are capable of dividing. You wouldn't necessarily want some osteocyte that was hanging out here in the middle of the matrix to divide, because where would it go and what would it do? Right? This is such a big, solid, crystalline matrix. These cells don't have any wiggle room to move around. That's why I need those long processes to be able to communicate with their neighbors and make sure they get the oxygen and the nutrients that they need, because there's no place for them to go. Awesome. Questions on that? All right, spectacular. That is, I believe, since we already looked at it once, let's cheat and look at it again. Oh, I guess that's the one thing we didn't talk about. We'll do that in a minute. We didn't talk about the red bone marrow and the yellow bone marrow, but other than that, I think we've got everything. Spongy bone, just the trabeculae, interstitial lamellae. Yeah, all right, excellent. Everything else there looks good. Perfect. All right, so with that, we are done with our histology. Again, like I said, not nearly as much as we were responsible for on the last exam. But when we talk about bones, there are also, again, the key here is 6. Structural classifications of bone. Your book does describe all 6, although your book tends to emphasize just 4 of them and then kind of mentions the other 2. And again, it does it for a reason, and so that's perfectly fine. But we want to make sure that we identify all 6 types. So let's start easy. What are those 4? Let's start with the 4 more common structural classifications of how we identify the bones of your body. Give me one of the classifications. If I were to reach into your body and pull out a bone and say, what type of bone would this? What would one of the possible correct answers be?
Shape.
Yeah, so yeah, based on shape, exactly.
Long bone.
Long bones, excellent. And of course, how do you define a long bone?
It's
If it's
longer
long.
than it is wide.
Yeah, exactly, right, exactly. A long bone is long, a tall building is tall, but you've absolutely got the right idea. Basically, it is a thin, narrow, right, elongated bone. One of the interesting things about the long bone is they're only found in the appendages. In the arms and the legs. Excellent. However, are all the bones of our appendages long bones?
No.
No. Oh, I'm asking the question, so the obvious answer is no. What are the other types found in the appendages?
Short bone.
Say again?
Short bone.
Excellent. How do we define short bones? Yes, they're short. How else would you describe them? Well, typically they're small block-shaped bones. Where are they found in the appendages?
Carpals,
The
metacarpals,
carpals, where are those?
hands,
Yep. Here in the wrist, we got our carpals. And anywhere else?
top of your foot,
Yeah, tarsals. Right.
tarsals,
Yeah,
metatarsals.
exactly. Exactly. Well, not the metatarsals. Metatarsals are long bones, but you are correct in that the tarsals and the carpals are our short bones, small block-shaped bones. What else is there? Give me another structural classification.
Regular.
Excellent. Regular. We'll get back to that one in a second. Let's cheat first. I like that one. That is absolutely one. But before we do that, give me a third.
Flat.
Flat. There we go. How do we define flat bones?
Flat.
Yes, the building is tall because it is tall. Excellent. These are also narrow bones, but typically they're narrow, broad bones. So they're thin, broad bones. And there's one other key characteristic to a flat bone. Not only are they broad and thin, but what else?
Smooth.
Say again?
Smooth.
True, although you could argue all of these are smooth on the outer surface. Give me an example of a flat bone.
One of the bones in your
Mostly.
skull.
Yeah, so many of those of the skull are. And what do you notice about, especially with someone like me who doesn't have hair, what do you notice about the shape of these broad, flat bones?
Curved.
Yeah, so a flat bone typically is narrow, broad, and curved bones. Now, the reason you are absolutely correct, irregular is one of our 4 main types that we were talking about. But the reason that I wanted to jump ahead of it is because as we've been struggling with our definitions, like a long bone is long and a short bone is short and a flat bone is flat, when it comes to defining our irregular bones, really the easiest way to do that is to say that they are not long, short, or flat. So that kind of is how irregular bones are identified. So we have these 4 main classifications. And with these 4 main classifications, this is where almost all of the 206 named bones fall into one of these 4 categories. And so these are the ones that are mostly emphasized. Of course, there's that pesky word, almost, because there are 2 other types of structural classifications of bones. One of those is a type called sesamoid. These sesamoid bones are called sesamoid bones because, again, most are the size and shape of sesame seeds. These sesamoid bones are typically found in the tendons of joints. How many joints do you have in the body?
600.
Yeah, well, I don't know if that's right or not, but it's a lot.
Neither do I. It's a lot.
It's a lot. Yeah, a lot. There's a lot of them, absolutely. And there are probably a dozen different places these sesamoid bones can be found. Some of them can be found in the wrist. Many of them are found in the ankle, right? Professional bull riders will get these on the tendons on the inside of their thighs from the use there. Basically, what these do is they help to adjust the angle of the tendon to make that tendon more efficient. So different people who do different activities will get these in different locations. So some of this can be from use, some can be genetic, right? They can be more likely to occur in the wrist for some people than others. But is everybody going to have the exact same number in the exact same location?
No.
Exactly. So that's why we said most of these are unnamed, because different people have them in different locations. Remember, one of the things we talked about is everybody has far more than 206 named bones in their body. Of course, notice there's that pesky word most again. Notice also I said most are the size and shape of sesame seeds. However, there are two that almost everybody has. There's a very, very, very small, rarely do not people not have two of these, but most people will have two of these that are very, very large in a very special location. And because of that, there are a couple of these sesamoid bones that are actually given specific names. And what is that big, most obvious of the sesamoid bones that people have?
Patella.
There you go. Chorus, help me out, right? I'm not as sophisticated as you are. What the heck is the patella?
Kneecap.
There you go. Reach down and wiggle your kneecap. All right, everybody here have two of them? There are some people that will only have one. One won't form. So rarely there are some people who have that where one doesn't form. But most people have two, and if you can reach down and wiggle that kneecap, that is your patella. Lastly, there are bones known as wormian bones. These wormian bones are only found in the skull, and more specifically in the joints of the skull. Because of that, the joints of the skull are known as sutures. These are also sometimes referred to as sutural bones. Let's talk a little bit about a suture, and I'll cheat a little bit by drawing this way. Here's, let's say, a piece of your skull. And on that piece of your skull, the sutures are typically these very undulating joints that hold the bones together very, very tightly. Now, even with all this undulation, we can still very clearly tell what is bone A and what is bone B, right? So if I took my highlighter and said, right here, A or B, what would you say?
B.
Right here, A or B?
A.
A, right? Exactly. But inside these sutures, many of them are the size and shape of a grain of rice, but some of them can actually be as large as a $50. Within these sutures, a small piece of bone can form. Notice this small piece of bone that forms is not a part of A, is not a part of B. It's its own individual bone that forms there inside of that suture. And do we all have the exact same number of them in the exact same spaces?
No.
No. So again, all of us have random numbers of these wormian or sutural bones, so we can't give them specific names. Most people have dozens of them. So like I said, while we all have 206 name bones, all of us have far, far more than 206 bones in our body. And those are the 6 types. So notice as we come here, here we see the pretty pictures. We see a flat bone like the one of our skull. We see a long bone like our humerus. We see the short bones like the carpals in our wrist. We see an irregular bone like one of our vertebrae. Here we see that sutural bone. Notice this is a relatively big one forming within that suture, or the wormian bone. And then here we see the best example of a sesamoid bone, that patella. So those are the 6 structural classifications of the bones of our body.
And that sutural, go ahead.
I was just going to ask if any of the sutural bones are named.
No, there isn't one that is consistently found in everybody. So no, there are no named sutural bones that I'm aware of. No, yeah. So there are no named sutural bones. Yeah.
And they're just like how you get a suture when you like getting sewed back up from a surgery or something like that. Is that basically what the sutural bone does?
No, although I get where you would get that from the meaning, but no, they get their name simply because these joints between the bones of our skull, these unmoving joints of our skull, are what are known as sutures, and these occur within the sutures. Why they occur isn't fully understood. If you think about it, one of the things we know about the skull is at birth, the sutures of the skull are not fully formed, right? You have to be careful about how you hold the baby's head. Babies have that fontanelles because the little fountain, soft spot on the top of their head because the sutures haven't fully formed. So as the bones grow together, these pieces of bone form inside of those sutures. And again, why it happens isn't fully understood. Why it's so random in people is not understood. Because like I said, why is one of those questions that can be really hard to answer sometimes in anatomy and physiology.
Isn't because the brain side, like the brain growing?
Well, okay, yes.
So it doesn't like.
Well, that's part of it. Also, that baby has to pass through the vaginal canal. So it makes sense why the sutures are not formed together. That makes sense from a biological standpoint. But why these extra bones form when they do grow together, that we don't fully understand. But yeah, no, you're right. That the massive growth of the brain, the massive growth of the body, the ability to pass through the vaginal canal and through the pelvis, those are all reasons why it's useful to have a head that's not fully formed, or the bones of the head not fully formed yet. But why these sutural bones form is not understood. All right. Questions on that? All right, how are we on time? Doing excellent. Perfect. Excellent. So while we've talked about these 6 structural classifications, we've already kind of hinted at there's some important anatomy for these that we need to understand. So let's start with the long bone, because the long bone is the most challenging with the most anatomy on it. So let's take a look at a picture of a long bone here. And because it gives me an excuse to draw another dino bone, let's go ahead and do that. We are going to talk about the anatomy of the long bone. Again, as we have talked about, as you guys mentioned, a long bone is a narrow bone that is longer than it is wide. But really, there's another key characteristic to it. It has a narrow shaft region, and then it has basically 2 enlarged head regions at the end. Of course, knowing how much anatomists hate us, are we going to get away with terms like head and shaft?
No.
No, of course not. So what is the name for the narrow shaft, the appropriate anatomical term for the shaft-like region of a long bone?
Diaphysis.
There you go. And again, remember, I don't care how you pronounce these things as long as you spell them correctly on the exam. And of course, how many diaphyses does a long bone have?
1.
Just 1. Excellent. And then what would the name then be for the head regions?
Epiphysis.
There you go. Epiphysis. And of course, how many do we have there?
2.
2 epiphyses. So we need a way to tell them apart. How can we tell the 2 epiphyses apart from each other?
Distal and proximal.
Right. Remember, one of the things we know about the long bones is they're only found in the appendages. And one of the primary ways we relate things in the appendages is by their relationship to the core of the body. So this one could be further from the core of the body, so it would, of course, be the distal epiphysis. And that would make this one over here the proximal epiphysis. Excellent. So 2 head regions, 1 shaft region, 2 epiphyses, 1 diaphysis. Now, we've already talked about some other anatomy that we're aware of. As we know, the center of the diaphysis is hollow. Here we have this inner region inside of the diaphysis. And what was one of those terms we learned before for an inner region?
Look, lumen.
Think of it in terms of the hair. The hair had a chewy nougat center and a candy-coated outer shell.
The medulla.
Yeah. So in here we have the medullary cavity. And what does this medullary cavity contain inside of it?
Marrow.
Bone marrow, excellent.
Bone marrow.
Now, well, in an adolescent, it is going to contain red bone marrow. In a fetus, developing fetus, it's going to contain red bone marrow. But as we age, as we mature, that red bone marrow collects a massive amount of adipose. And basically, in an adult, is going to contain yellow bone marrow. So in an adult, this medullary cavity is the main place where you're going to find the yellow bone marrow. Now let's cheat and do this a little bit. We learned a little something else about our medullary cavity. We now know the medullary cavity is going to be lined with a little bit of spongy bone. And of course, on top of that will be our endosteum. So let's make a thin blue line. Oops, I wanted it to be like that.
And that wraps that entire cavity.
Lines the entire cavity, yes.
Yeah, it lines the entire. Yeah.
And as we know, that is going to house the osteoblasts and osteogenic cells. Excellent. Since we're doing it, we also know on the outer surface, there's going to be a dense irregular connective tissue. Oops. Ran out of mouse pad and ended up hitting my computer. Let's try this again. Lining the outer surface is a dense irregular connective tissue. And of course, what do we call that?
Periosteum.
There you go. Which is going to both house immature cells, like the endosteum. But remember, it also stabilizes blood vessels, attaches tendons and ligaments, protects the bone, and I feel like there's one more thing. Houses the immature cells, stabilizes blood vessels, attachment point for tendon, ligaments, and proteins. I think that's good. Why do I feel like I'm forgetting something? I guess that's it. All right, excellent. All right, and then of course, a thick layer of compact bone. And that's pretty much the anatomy of our diaphysis. Questions on that? All right, then let's talk about our epiphyses. Our epiphyses are lined by a very thin layer of compact bone. And there's no medullary cavity. Instead, it is completely filled with spongy bone. Right, those are regular points and processes of our spongy bone, those trabeculae. Now, of course, those are going to be those trabeculae are lined with an endosteum. So there is indeed going to be an endosteum lining this space. And what are we going to find inside the spaces? Yeah, not just cells, not just any of those cells, but those hemocytoblasts, because here in the epiphyses is where in a mature adult we find our red bone marrow. After all, as we get big, we need to lighten up the load of our bones by getting rid of that red bone marrow and filling it with adipose instead. But even as adults, do we still need to be able to make red blood cells and white blood cells and platelets?
Yeah.
Yeah, absolutely. And so we have to have red bone marrow. So in an adult, red bone marrow is primarily found inside of the compact bone of the epiphyses, and then as we'll also see inside of flat bones and short bones and irregular bones. Has anyone here ever donated bone marrow before?
I've heard it hurts really bad.
If you have, you are truly a rock star, and I commend you for it. Heck, I'd even probably give you extra credit for it because, as Brian pointed out, it is an incredibly painful process. Basically, they take a huge, massive bore needle and they insert it either into your hip bone or into the head of your femur so they can extract a little bit of red bone marrow. And even though it's a huge bore needle, is one insertion of that needle going to be enough to pull out enough bone marrow to provide the cells necessary to help someone survive and live?
No.
No. 6, 8, 12 times they have to insert that needle into the bone, suck out a little bit of that marrow, and collect it. Yeah, they give you a local, but I am told it hurts like hell. It is a brutally process, but it is an amazing gift for an individual to give to be able to do that. But that's why they have to do that, because they have to go into those locations where there's a large amount of it to be able to extract what is necessary. So here inside of the spaces is where you're going to find your red bone marrow, just filling in these spaces here. Now, there's one other important characteristic about our epiphyses. Our epiphyses is typically where one bone forms a joint with another bone. And we want to protect the bone while it's doing that. So what would be useful to put on the outer surface of this bone to provide that necessary protection?
Hyaline cartilage.
Exactly, right? So here on the outer surface of our epiphyses, we are going to have some hyaline cartilage. Obviously, hyaline cartilage is the type that lines the surface. Of course, it's not the only place we find hyaline cartilage here, so it's also given a name based on its location, or really on its job. It's called articular cartilage because it forms the articulations. Articulations, just a fancy way of saying joint. So our hyaline cartilage, or this articular cartilage, lines the outer surface of our epiphysis. And that is the basic anatomy of our epiphyses. Now notice the anatomy of our epiphysis and the anatomy of our diaphysis is dynamically different. So not surprisingly, there is a transition area. That transition region, basically between the epiphysis and the diaphysis, is what is commonly referred to as the neck of the bone. But are we going to get away with a term like neck when we're talking about this for the bone, for our long bones?
Of course not.
No, of course not. So what would we call this region?
Metaphysis.
There you go, exactly. It's the metaphysis. And of course, we're again going to have two of them. So we'll have a distal and a proximal metaphysis. So two metaphyses, just like there are two epiphyses. Obviously, this is the transition from the medullary cavity to the solid spongy bone that is found here as well. But there is something else interesting found in the metaphyses. In the metaphysis of our adolescents, our babies, there is a chunk of hyaline cartilage. This chunk of hyaline cartilage is what is known as, and again, it's in the metaphysis, but it's called the epiphyseal plate. Anyone know what this epiphyseal plate is for?
Growth plate.
Yeah, this is what we commonly refer to as the growth plate. And what does the growth plate allow the bone to do?
Grow and elongate.
Yeah, exactly. Allows the bone to grow in length. And this epiphyseal plate, as I mentioned, is formed from hyaline cartilage. So notice not only do we have hyaline cartilage on the outside of the epiphysis, we have hyaline cartilage basically attaching the epiphysis to the diaphysis, known as the growth plate. Right? Because how many people here are the same size as they were when they were born? Anyone? If so, I feel very, very sorry for your mom. Excellent. No, we've all grown massively since we were born. And the reason we've been able to get taller is because of this epiphyseal plate. However, how many of you are still getting taller? Anyone
None.
still getting taller? I'm still growing, more outward than upward. Anyone still growing upward? Why not? Why isn't anyone getting taller? Why aren't people like...
Stops functioning at a certain point?
Sort of, yeah, absolutely, right? Why aren't people like Barry Bonds 10 feet tall? After all, he allegedly took growth hormones, which would cause the bones to grow and get longer.
We'll lose structural integrity.
Not so much that we lose structural integrity, but what actually happens as we mature at the end of puberty, what actually ends up happening is that epiphyseal plate fuses and basically is completely ossified. And when it's completely ossified, it basically becomes a thin line of compact bone. Once it becomes that thin line of compact bone, it can't grow anymore in length. Your bones are as long as you're going to get. So Barry Bones' head got bigger, right? His knees got weaker, but he didn't get any taller, allegedly. All right. When it ossifies and becomes that thin line of compact bone, you're as tall as you're ever going to get. And in the bone, we now call that structure the epiphyseal line. So if you just randomly found some leg bone on the street and you were curious who might have lost it, one of the things you can do is to cut that bone in half and look in this metaphysis. Yeah, I mean, you could think, well, the size will tell me whether it's an adult or a kid. But there are plenty of adults who are 5'1", and there, especially if you watch Sports Center, plenty of 8-year-olds that are 6'3", playing basketball with kids half their size, just dribbling them into the ground. So size isn't necessarily the only way you're going to be able to tell. An easy way to get a general idea of the age is to look in the metaphysis. If you see that thin line of hyaline cartilage, you know this is typically someone who is not reached their full sexual maturity yet. So typically someone under 25 years of age, because usually 18 to 25 is when we end puberty, when that epiphyseal plate closes. Whereas if it's closed, then it's typically someone who's reached their sexual maturity and would be someone typically 25 or older.
Is there a chemical or a signal or anything like that that basically tells the bone, hey, bruh, stop?
Yep, exactly. And like I said, part of the learning of all this anatomy, once we learn the structures, we can talk to physiology. So are we going to talk about how bones grow in length and why they stop growing in length? Absolutely. I guarantee it. But for right now, we just have to know the structures, then we'll figure out how they form.
Professor,
Yes.
can you save this image on Canvas, please?
Sure, I can do that. I think we're done too. I'll go ahead and save it now, but let's look at the pretty picture from the textbook and see if there's anything we have to come back and add. All right, so here we have our long bone. As we mentioned, we have the shaft region, which is the diaphysis, the two epiphyses that we are going to distinguish by distal and proximal. Notice also for many bones, unlike my dino bone that I've been drawing, that dinosaur bone, often the proximal and distal epiphyses will look different from each other. It's one of the ways we'll be able to identify the bones, but they don't have to be for us to be able to talk about the anatomy. We know that there is articular cartilage on the ends. And what kind of tissue is that articular cartilage again?
Hyaline.
Hyaline cartilage, excellent. And we also have the metaphyses, which are these transition regions from the diaphysis to the metaphysis, what we commonly refer to as the neck regions. In there, we have either the epiphyseal plate or the epiphyseal line. Notice this one's a thin line of compact bone, so this one's an epiphyseal line. Whereas if this was a larger chunk of hyaline cartilage, then that would be an epiphyseal plate. So we can tell that this bone came from someone who was typically over 25. Again, we have the periosteum on the outer surface. What type of tissue is that again?
Dense irregular connective.
Dense irregular. And like I said, basically 4 things it does. Houses the immature cells, stabilizes the blood vessels, right? Provides protection for the bone, and acts as an attachment point for tendons and ligaments. There you go. Our medullary cavity is where we contain our yellow bone marrow. This picture doesn't show it, but remember that other picture did. There is going to be that thin layer of spongy bone on the inner surface. And typically in a mature adult, it's going to contain yellow bone marrow. And it'll be lined with our endosteum. And again, we don't need protection. We can't attach anything on the inside. So really, it primarily just houses those immature cells and the osteoclasts. And there you go. We've got it all. Excellent. I think I saved it, but let's cheat and go back and save it one more time just to make sure. All right. Excellent. Questions on the anatomy of the long bone? All right, perfect. So that is the more challenging one. The good news is, actually, you don't stay here. Let's do that. The long bone is the one with the most elaborate anatomy. So basically, the anatomy of the short, flat, and irregular bones is essentially the same. Let's go ahead and draw a box-like structure. Basically, if we take a cross section through a flat bone, a short bone, and an irregular bone, it is going to be just like with the compact bone. I mean, just like with the long bone. I don't remember what color we used. Was it blue? Maybe. Periosteum on the outer surface. And that periosteum sits on top of a thin layer of compact bone. Now, this thin layer of compact bone, you could also almost argue, is like a candy-coated shell on the outer surface of these bones. And what would the term we would give for such a structure be?
Cuticle.
Close.
Cortex.
It wasn't cuticle,
Cortex.
but cortex. There you go. So this is the cortex of the bone, this thin layer of compact bone on the outer surface. And this is, of course, the cortex down here as well. Oops. There we go. And then in between, and I'm just going to draw them as solid lines because it's simpler, we're going to have all of the irregular points and processes that are the trabeculae. Right? No medullary cavity. For this one here. It's still going to be lined with an endosteum that will house the immature cells and stuff like that. So here and here and here. So all of this is going to be aligned with our endosteum housing the immature cells. And then, of course, in the spaces, it contains our red bone marrow. So our short, flat, and irregular bones are also where we find our bone marrow. So again, all these spaces would contain our bone marrow. And that's it. The anatomy of our short, flat, and irregular bones is pretty simple, except for one small thing. What's the number one thing you need to learn in this class?
Anatomists hate us.
Exactly. Anatomists hates you. And they find lots and lots of ways to describe this. This is the anatomy for short bones, flat bones, and irregular bones. Same for all of them. They all have the same general organization. However, for some reason, and I honestly, all kidding aside, have no idea why, but in flat bones only, and only in flat bones, they give a special name to the trabeculae in the center of our compact bone. And that special name they give is diploe. And hold on, I got to cheat because I got to give the little umlaut on top of the E. There you go. I'm sure there's a way I can do that with my keyboard, but I don't care to. It's more fun to draw the dots. Why they're called diploe in flat bones and no other bones, I honestly have no idea. But if you were looking at a flat bone, the trabeculae of the flat bone are referred to as diploe. So again, let's look at the pretty picture from your textbook. Here it is. Notice, taken from the skull. Here we have our outer layer of the compact bone. Here we have the trabeculae. We know there's a periosteum on the outer surface. We know there's an endosteum line in the inner surface. We know red bone marrow is found in here. And again, that is the same for all of our bones. But for whatever reason, in a flat bone and a flat bone only, these trabeculae are referred to as diploe. All righty. And there is not just the microscopic, but the gross anatomy of our different types of bones. Questions on that?
Nope.
All right, excellent. With that, we can now move on to the lab materials that we need to talk about. So actually, let's do this. Let's go ahead and take one last break right now. And then when we come back from the break, we will talk about what you're going to be responsible for, obviously on the exam, because that's what you care about, is how you're going to be successful on the exam, but the way we're going to prepare for that by our group presentations. Again, nobody is being graded on their presentation skills. This is about as chill of a presentation as you could possibly be. The whole point of this isn't to force you to do something you're not comfortable with. It's just to make this a more active learning process. If we were in the classroom, you would be standing up in front of the class and doing this. We would be doing it there as well. So I'm not penalizing you for this being online. This is what I found to be the best way to do this. Because rather than me holding up 206 bones and pointing out all the different bits and pieces of it, I find it much more interactive and more fun if we do it together as a group. So that's what that's going to be. So again, nobody should be panicked about this. It's going to be totally chill. You're not being great on your presentation skill. You don't have to memorize your presentation. So if you want to sit there and read it off a piece of paper the whole time, that is totally fine. So again, it's totally chill. But before we talk about it and get into what we're going to do, let's take one more break. We'll take a full 15-minute break, come back at, it'll be 10:37, and at 10:37, we will talk lab stuff. And make sure you come back, because when we come back is when I'm forming the groups. All right, see you in 15 minutes. What we are going to do now is talk about our bones and bone features. Here we have a nice handout chart from your textbook. All the bones have things that are called features on them. And when you look on your color atlas, like you are going to be want to do, when you look at a particular bone, you will see that many of their bone features are listed. Now, again, some of this is a little hopefully obvious, but let's go ahead and make sure we're doing this. When we talk about a bone feature, it is a characteristic of the bone. Hopefully that seems and sounds really obvious, because hopefully it is, right? Now, every single bone is going to have bone features on them. Does that mean you are responsible for every single bone feature of every single bone?
Yes.
No, right? I'm mean, but I'm not that mean, right? Is there a way to know how many bone features you are responsible for? Let's say, for instance, we took a bone like the frontal bone. The frontal bone, which happens to be the front bone of your skull, how many bone features do you think it has total?
5.
3.
Yeah, 3, 5, might be as many as a dozen. How many are you going to be responsible for on this exam? Excellent, Veng. You're absolutely correct. How do you know that? Okay, that would you guessed correctly, which is awesome. Was there anybody else who knew it was one and didn't guess?
Maybe because you only put feature instead of features.
Also not a bad guess. However, being the smart, sophisticated students that you are, what hopefully you have started to look at already is your handouts for this section. Not only is there a skeletal system histology handout, but notice there's also a bone and bone features handout. And on that bone and bone features, there's a bit of an introduction. There are the bones of the skull. And then on the second page are the bone features you're responsible for. So we were able to guess and figure out that there's one bone feature we're responsible for the frontal bone. Anyone able to guess what that specific bone feature might be?
Frontal
Front
sinus.
of the limb,
There
frontal
you
sinus.
go, frontal sinus. How many bone features do you need to know for the ethmoid?
7.
How many for the temporal bone?
9.
9. How many for the palatine bone?
1.
How many for the vomer?
0.
0. Does that mean you don't need to know the vomer? What do you think? Are you going to be responsible for identifying the vomer bone on this exam? Yes or no?
Yes.
Yes. The bone, yes. But none of its bone features. Does that mean it doesn't have bone features?
No.
No. Every bone has bone features. But in this case, you're not going to be responsible for any of the bone features of the vomer. Every single bone on your list, let me rephrase that. Every single of your 206 named bones in your body have bone features, numerous bone features. But for the exam, the only ones you're going to be responsible for are the ones on this list. All right. Now, again, these bone features, as we've talked about, are given specific names. These are characteristics of the bone. Some of them are things that stick out. Some of them things are going to be attachment points for muscles. Some of them are going to be points for joint formation, indentations, holes through them. And for the most part, there are rules on how these things are named. Right? There are rules. So on an exam, if you identify something as a trochanter or you identify something as a tuberosity, do those two things mean the same thing? Is the term trochanter and tuberosity, do you think, interchangeable?
No.
No. So for instance, on the exam, if I point to this bone feature right here and you identify it as the ischial trochanter, you're not getting that correct because it is instead the ischial tuberosity, right? If you were to identify this as the greater ramus or something like that, that would be wrong. It is the greater trochanter. So these names have meanings, there's definitions. A hole is a foramen, an indentation is a fossa. Your book has a nice table of the definitions of these things, and knowing the definitions is definitely something that will be useful in helping you to be able to interpret. Let's see, is that a big round attachment point or is it a small rough attachment point, right? Using these and understanding these rules are something that is important. All right. So for the exam, you are going to be responsible for bone features. You are also going to be responsible for skeletal features. A skeletal feature is a characteristic that is formed by 2 or more bones. So right here, we have, right, what I commonly refer to as my cheekbone. You don't actually have a cheekbone. What you actually have there is an arch-like structure of bone. And that arch-like structure of bone is made up of two bones coming together. So notice if I'm just holding one bone in my hand, this hook is a bone feature of this bone, but it's not forming an arch. Here I've got my second bone. It has a bone feature. It has a hook and it has a name, but I don't have an arch. Only when I take the two together and put them together do I get the arch. So you are going to be responsible for skeletal features. You obviously need to know them by name, but you also need to know the bones and bone features that form them. And I will ask that on the exam. I could have that arch-like structure and I could put an arrow and I could ask you to identify the skeletal feature, or I could point at that same arch and ask you to identify the bones and bone features that form them. Now, probably the most complicated structure in our skeleton is the skull. So not surprisingly, all of the skeletal features of the skull we call skull features. So skull features are basically just skeletal features of the skull, right? That is essentially what they are. However, if we want to be more specific in our definition, it is a feature of the skull that is made up of two or more bones. So on this exam, you are going to be responsible for bones, you are going to be responsible for skull features, you are going to be responsible for skeletal features. To help us to master all of this material, you are going to be doing group presentations. I will be in a moment dividing you all into groups. And when I divide you into groups, as you will now see available in your modules, under the group lab activities is a list of the skeletal system group presentations for the axial skeleton. That is where we're starting with our presentations on Thursday and finishing them on Friday. You will be randomly assigned by Confer Zoom into one of 8 groups. And these 8 groups are going to be presenting this material. We're going to go in order from that student info sheet, the study guide. So notice the first thing on that study guide is the frontal bone. So if you're in group 1, you're doing the very first presentation. And we're just going to go down the list of stuff that way. To help you with this, I am going to give you, once we're done with this discussion today, the rest of the class time today, which is going to be close to an hour and a half by the time we're done talking, to work in your groups here in class to discuss how you want to present this. I will also be setting up discussion boards for you. That did not work at all. Discussion boards. So in the discussion board, it hasn't been set up yet, but as soon as I add you into groups, I will then be adding you to the discussion groups. And so you will have your own discussion board where you can communicate asynchronously. As a group, you are welcome and encouraged to exchange contact information, emails, phone numbers if you want a text group. I've had groups set up Discord groups before if you want to do that. Now that the world is coming back to normal, if you want to go meet at Starbucks or Chipotle, you're welcome to go do that as well. Don't do it here in Roseville because apparently it's dangerous, but you can meet other places to be able to do that. Again, it's not required though. If because of your work schedules, because of your life schedules, if you're not capable of meeting up, then you can use the discussion board to do comment and work on this material asynchronously so that I can do my stuff and put it on that discussion board and you can do your stuff and we can be prepared for our presentations together. Okay, so you're encouraged to work synchronously with this. You have the rest of the class time today to work synchronously on this, but you're not required to meet outside of class time. So that is what the discussion board is for. If you can't meet outside, or you're too busy, or you don't want to share your phone number with anybody, then use the discussion board to be able to communicate with people that way. Now, let's talk about what you are going to be responsible for. As I mentioned, for these group presentations, you are not being graded on your presentation skills. You do not have to be prepared. Like I said, if you want to read it off of a piece of paper while you're doing it, that is perfectly fine. When you're presenting, the entire group will be on camera. However, much like we're doing now, you are going to have your slides up here, and you're just going to be a tiny box in the corner like I am. No one's going to be looking at you anyway. But so you do need to be on camera for this presentation. Obviously, you want to introduce yourselves, because not everybody's necessarily had a chance to meet each other. So make sure you give your names, and you're going to be on camera for this while you're doing these presentations. But like I said, you're not being graded on your presentation skills. So let's talk about what you are going to be responsible for. Obviously, the number one thing you're going to be doing is presenting your bone. How are you going to do that? However you like. If you happen to have a skeleton in your closet, you are welcome to use that. However, most people don't, or at least most people don't want to share them. So you will notice the other thing that I've made available for you here in the modules under the group lag activities are pictures that I thought were good for these presentations. So I have made a handout here, some PDFs that show some images of the skull from different points of view, from the outside, from the inside. You are welcome to use these if you would like, but you're not required. If you want to find your own pictures, if you want to put together a, like a presentation or something like that with slides, you're welcome and encouraged to do that. With this PDF, what I find best is, again, if you want to make a slide and do it that way, that's fine. If you want to do it real time, where you want to point and go, this is the inferior nasal concha, right? And everybody who's an Oz, then that's fine too. But the point is, your job is going to be to present the bone. That is the first thing you're going to need to do. The second thing you need to do is present your bone features. And again, for the bone features, you want to focus on the study guide. Every single bone on your list is going to have more bone features than you're responsible for. So if you're that first group presenting that frontal bone, do you want to spend 15 minutes telling me about all of the bone features of the frontal bone? No, because how many of those bone features are you responsible for?
One.
One. And while I am a big fan of knowledge for knowledge's sake, the point of this is for us to prepare successfully for the exam. Now, when our first group presents the frontal sinus of the frontal bone, is everybody going to instantly have it mastered and be successful on it on the exam?
No.
No, but the point is to work together to expose ourselves to this information so that when you're studying this on your own, using your own resources, like your histology atlas, like the PAL program on mastering A&P, and any of the other, there's a great link to a bones section on in the study area where that can go, and things along those lines like that. So again, present your bone features, focus on the study guide. Also, and here's the key, for many of those things, you don't need to know their function. Now, again, Some of them may be a little bit obvious. When we get to the temporal bone, we will talk about the temporal bone has a hole in it known as the carotid canal. What do you think the function of the carotid canal is?
For the artery?
Yeah, carotid artery passes through that, right? Again, the name kind of gives it away. However, am I going to ask you the function of the carotid canal on the exam? No. The one exception is for articulating surfaces. Articulating surfaces is where one bone forms a joint with another bone. And joints are part of this section. So on the exam, I could show you a bone feature of bone 1 and ask you to identify this bone and bone feature. But I could also ask at this point at this bone and bone feature and ask you for the bone and bone feature it articulates with. That's actually a lot of the skeletal review. The skeletal review, remember I told you, is knowing how bones relate to each other. So if you're showing me an articulating surface, it might be useful for the class if you tell me what bone and bone feature it attaches to. So you are responsible for articulating surfaces. So it'll be useful if you tell us what bone and bone feature it articulates with. All right, helpful, useful if you can do that. Again, you're not going to lose points if you do this. You're not getting graded on your presentation skills or your knowledge. If you mess up, you're not going to lose points. Everybody, as long as you're here, camera on, and you're active in the learning process, you're going to get full credit for doing this. Like I said, it is as simple as that. All right, so you're going to present the bone and the bone features using either the images I've provided or images that you get together on your own. Okay, so that's that. Another thing that is going to be important is you are going to want to present the bones from different points of view. Remember, one of the things we talked about is that we need to get used to looking at the body from different points of view. And again, this section is normally the easiest and the most fun because everybody loves the bones. But they love the bones because they get to pick them up and play with them. Taking a bone and looking at it and twisting it around is the best way to learn that. We're not getting the opportunity to do that. So this section is much more challenging than it normally would be. I've tried to provide multiple images of the bone from multiple points of view so that you can see it and recognize it easily. And again, so you'll want to present people with the bones from different points of view as well. And including for the skull, there are some bones that you need to know disarticulated. Anyone know what disarticulated means?
They don't articulate. They're
Yeah.
fused.
When they're separated from the skull. And in particular, those bones are the sphenoid, the ethmoid, the mandible, the maxilla, temporal, sphenoid, ethmoid, mandible, maxilla, and palatine. Those 6 bones, if you are presenting one of those 6 bones, you need to give it to us both in an intact skull and disarticulated. And if you will notice, so for here, for instance, you would show me the sphenoid in an intact skull or the ethmoid in an intact skull. And then notice my pictures have also got it disarticulated as well. Here's the sphenoid disarticulated. Here's the temporal bone disarticulated, maxilla disarticulated, mandible disarticulated, and the palatine disarticulated. So again, I've given you these pictures. You're not required to use it, but I've tried to make it easy. And again, could some of these pictures be on your exam? Absolutely. Am I going to am I going to contain myself to just these images for the exam? Absolutely not. Because your goal isn't to memorize these pictures. Your goal is to understand this material. So this, I did a quick Google search to find all of those. That was what was useful for me. You are welcome to do that however you want as well. So present your bone. Make sure you're on camera. Give us the bones and bone features you're responsible for. Present the bones from different points of view, especially if you're presenting one of these skull bones. You are going to present it in disarticulated as well as in an intact skull. And the other important thing to remember is that for paired bones, and what do I mean by paired bones?
Are there two of them? So there's
Yeah.
like a left and a right.
Exactly. If you have a paired bone, that means there's two of them. There's a left and a right. And if you have a left and a right, do we need to be able to tell which is which? Yeah. If your bone happens to be a paired bone, tell us how to tell the difference between left and right. All right. Obviously, you've only got one sphenoid. You don't have to worry about left and right. But you do have two humeruses. So when you're presenting the humerus, you need to be able to tell us what is left and what is right. So that's going to be our goal. This is what you're responsible for these presentations. And like I mentioned, we are starting on Thursday. So if we come back here, like I mentioned, know your bones and bone features. You have your study guide that is going to show them, and we are going to go in order of those as well. As I mentioned, our game plan is going to change starting Thursday. In lecture, we are going to be focusing on the processes of bone homeostasis. How you make a bone, how you grow a bone, how you maintain a bone, how you repair a bone, all of the things going along with that. And then after I left lecture for that for a time, we are going to get together and work on our bone and bone feature list. Again, going right in order. Our goal for Thursday, because again, let's come back to our handouts. Our goal for Thursday is going to be to get through the skull. We are focusing on the axial skeleton first, and we're going to try to get through the skull. So our goal is going to be to get through the first three groups. And then I will help you by putting all of the stuff that they give us together, and together as a group, we will work on the skull features. And then on Friday, we'll do the rest of the axial skeleton. Also on Friday, I will assign you the appendicular bones that your group is going to be presenting. So you will be presenting twice. I guess we need to put that back on our list as well. We'll be presenting twice. The axial skeleton will be Thursday and Friday. And the appendicular will be Monday and Tuesday. All right. I'm purposely not giving you your appendicular bones yet, because for right now, for tomorrow, for the next 2 days, I want you focusing on the axial skeleton. Then you'll have the weekend to work on getting your presentations ready for the appendicular and learning your appendicular skeleton. All right. Any questions on the process or the game plan?
They'll be the same groups for
Yes.
axial.
Yeah, it's too complicated to break up the groups into different groups between them. And again, based on the number of students we have, you will either have 3 people in your group or 2 people in your group. And where did I started writing this down. Where did I put it? Excellent. So I was going through during the break. I have a way of randomly assigning you, but I haven't put you into your breakout rooms yet. So I wanted to make sure that nobody ran away while this was here. Oh, someone did run away.
Hey, so is everybody getting an individual bone, or is there a bone per group, or how's that?
So again, it is going to be, it depends on the group. So if we go back to our list, because hold on, I've lost somebody. So notice, can you guys see the list now?
Yeah.
Okay.
Yeah.
So if you're group 1, you have 3 bones. Now, again, notice the frontal bone only has 1 bone feature that you're responsible for. So it wouldn't necessarily be fair to have one person do the frontal, one person do the ethmoid, one person do the occipital. I mean, I guess if you decide that's how you want to break it up, that's, I guess, fine, but it's not necessary. Group 5, for instance, just has the thoracic vertebrae. So again, there is going to be some variation. So if what I find works best for most students, and again, it's your group, you may do it however you want to do it. But if in total, whether it's 3 bones or whether it's 1 bone, you have 12 bone features you're responsible for, well, and 3 people in your group, then that's 4 bone features for each person. But again, you as a group may decide to break it up however you want. Okay. You are there. How do I?
Are we also going to get in groups for the muscular and nervous system?
Muscular, maybe. So we'll see how things are going. Probably not for the nervous, but we and often for the muscle, I do switch it up because again, we may have lost some more people by then. All right, I'm going to go ahead and because I think all of our groups are still intact. One person disappeared from the list, but I should be able to add them back. All right, so any other questions on this process? Okay. Last thing I want to mention, I'm going to put you into your groups right now. Introduce yourselves, meet each other, contact information, whatever. But before you leave, I need to come into each group and I want to talk briefly with each group, individual group, about their specific bones and what they're going to be doing. So I do want to meet briefly with each group because each group has a couple challenges that I want to make sure that everybody is, that it would be a waste of time for me to tell everybody right now, but I want to do them with each group. So please stay in your group. Do not leave until I come to your group and talk. And then again, you guys have the rest of this class time to use this however you would like to. All right. Excellent. So get into your groups. I'm going to put you in your breakout rooms. I will bounce back and forth between the rooms. If you have any question, there is a way to kind of raise your hand or get my attention as I'm working my way through. So let's put everybody in their groups. And I think someone else just got scared and ran away. So hopefully we can figure out where everybody is and get them into their appropriate groups. So, all right, let's go ahead and give this a shot. Away we go.